enzymes.
Okay, thanks to the organizers for letting me present our work in this amazing venue, and I'm very happy also that I got a travel fellowship to come, so thanks for that as well. So first of all, I want to mention that this is the work of my first ever student, Maria, from Argentina, and it was a collaboration with my PhD supervisor, Diego Ferreiro in Buenos Aires, and Professor Peter Warnings in Rice University in the USA. So this work was recently published, so I'm also happy to be presenting this here for the first time. So before anyone here thinks that I'm going to talk about something related to psychology, I will explain what frustration means in, in this context. So if we have two magnets and they are energetically paired, then we don't have any conflict in this system. But if we want to add a third magnet in this configuration, then we cannot satisfy all the interactions at the same time, and then we say that this system is frustrated. So proteins, when they fold, they decrease the energy as they become structurally more similar to the native state. And this is because the interactions are cooperative, and random contacts within the polypeptide chain are less favorable than the native ones. So we have a huge bias towards the native state, and that's why the energy landscape looks like a funnel. But although the energy is minimized, not all the conflicts can be solved, and we always observe some energetic conflicts at the native states of proteins. And the reason why these conflicts have been kept there by evolution is because they should be useful for something, and that's the question we are trying to answer. So can we quantify these conflicts? Are they really important for protein function? So we have an algorithm for calculating and localizing frustration. It's very simple. So given a protein structure, we can define contacts given some bias, some threshold in distance. And by using an energy function, we can assign the native energy to all the contacts that we have in the protein structure. Then we can perturb these interactions by generating decoys or alternative configurations or identities. And for all these decoys, we can also measure the energy and calculate a distribution of energies and compare this distribution to the native interaction that we have in the protein structure. So by applying a very simple Z-score, we can classify these native interactions as being highly frustrated. In the case, most of our variants, they decrease the energy, as being neutral if the energy doesn't really move that much, or as being minimally frustrated if most of our variants are worse in energetic terms. So we have different types of frustration indexes according to how we construct these variants. So we can modify the identity of the residues that are in contact, or we can change the conformation a little bit, or we can aggregate all the interactions in single residues, and in that case we have the single residue level frustration index. So in our group, they have tried to understand this better for quite a few years already. So the first survey was on monomeric proteins, and when they did this, they observed that 40% of the interactions in these protein structures are minimally frustrated, so they are okay with the environment, but up to 10% of the interactions are in conflict with the local structure in which they are located. This is a pair distribution function plot. So here we have how are the density of contacts distributed according to each other in the protein structure. So the black line here is the distribution that comes because of the topology of the structure itself. And then we have classified the contacts as frustrated, minimally frustrated, or highly frustrated, and how they are distributed along the structure. So we can observe that compared to the overall distribution, the green and the red are enriched in some cases. So the minimally frustrated contacts form extended networks because we can see that they are over the black line at long distances. The neutral contacts are randomly distributed because they are below the black one, and the highly frustrated contacts tend to cluster at the surface. If we observe this better, also these red things are not randomly distributed in the protein structure, but they are clustered in specific places. They have been shown to be clustered around binding sites. I will show this pair distribution function quite a bit. So they are enriching binding sites and surface residues, and also it has been shown that these type of interactions are clustered and enriched around mobile residues in case of allosteric proteins. So we have this algorithm already available as a web server. You can put your favorite PDV ID code here or upload your structure, and in a couple of seconds you can have your frustratogram and observe how this is in your protein. So a lot of groups, since we published this, have been using this and trying to see how this is related to different functional aspects in protein structures and so on. So the thing is, why did we try to go further with this in protein enzymes? So the thing is that enzymes are very particular because they need to put together residues that can be quite distant in sequence, and they need to do it in a very precise way, and maybe these residues, they are not really compatible with each other in terms of energetics. So the rest of the structure needs to somehow hold them together in order for them to be happy, let's say. So the thing is that the hypothesis is that maybe the catalytic sites are enriching in conflicts, and the rest of the structure is kind of holding them there together. So if we see this qualitatively, here the catalytic residues are depicted in yellow, we can see that in some cases, in most of the cases, we can see that there are red lines around the catalytic sites, but the thing is this consistent? Is this something we can measure more better? So the thing is that we took all the non-redundant proteins in the catalytic site atlas that have solid structures. We have this number in total, and we calculated the frustration, and we calculated how the frustrated interactions are distributed around catalytic sites. So we observed that the catalytic residues are enriched themselves in energetic conflicts, and also in the second shell we can see an enrichment of this for monomers, homodimers, and this is all the others together, including these two. And this is true even if we take into account the buried or the exposed catalytic residues. Even this also stays true if we subdivide that data set according to all the EC classes. So far I have shown that catalytic sites are enriched in interactions that are conflictive, but we have seen this as a general thing. So can we isolate frustrating contributions to specific residues? And for this, we need to go to the level of protein families. So for this, we can calculate frustration for all the proteins that are present in a multiple sequence alignment, and then we define an alphabet of states, which is either the residues in this alignment are minimally frustrated, neutral, or highly frustrated, and then we just use the typical entropy information content measures to calculate the sequence information content as we do, and also we can do the same, but based on frustration states, and calculate the frustration logo and see how conserved the frustration state is along all the positions in a given sequence alignment. So we did this for beta-lactamases. Here, the catalytic residues are depicted with asterisks. The points, they are showing the sequence entropy, and here the bars, the color bars, are according to the frustration information content. So the higher the bar, the more conserved the state, and the color that contributes the most is the one that is most represented. So we can see that many of the catalytic sites, they are highly frustrated. For example, this one here, and so on. But not only those, and this is something that also was really surprising for us and very interesting, that also non-catalytic residues were holding high frustration information content. And for example, this residue here, when it's mutated, compromises the level of resistance to antibiotics. This one here is important, is one of the prolines that is isomerized for solving the fast phase in the folding of this protein. So the thing is, this can be maybe a bit beyond only protein enzymes, and we can go a bit farther. So can we use this tool, frustration, as a more general tool? And this is something that is not published yet, so it's the first time also we talk about it. So it's like, if we take into account protein families that are evolutionarily related, for example, globins, and we have hemoglobin that is composed of alpha and beta units, and these have a common ancestor, can we see how frustration changed over evolutionary time? So here we are having all the globins together, and this is the sequence logo, and this is the frustration logo. And then we separated the alpha and the beta chains and put them here. So this is the alpha subunit, and this is the beta subunit. Here, with the dashed lines in blue, we are showing regions in the alignments in where the frustration changes from comparing all the proteins together when we go to alpha and beta. For example, here we have, in the overall distribution, we have, like in this position, you can have a Q or a K, for example, and then the alphas only have the K, and the betas only have the Q, whereas the alphas have a highly frustrated site, and the betas, they don't have it. So we can say that maybe over evolution, we have changes in protein sequences that are reflected in the protein structures, and then we got the diversification in terms of energetic adaptations to have the specific function to the alpha compared to the beta. So to close up, protein enzymes, active sites are energetically frustrated in general. Energetic frustration in enzymes is conserved at the protein family level. Non-catalytic function-related residues can also be highly frustrated across protein families, and this is something that we can go farther in the future. And frustration patterns could be useful to track changes over evolutionary time and see if we can point out functional adaptations among paralog ortholog protein families. So just to acknowledge the rest of the people that work with us, so Maria was the leading author. This is another student we have now that is going to pursue the evolutionary part in collaboration with Cristina Orengo. If we are lucky to have everything going on. And this is the rest of the team. And of course, Professor Warnings that also was contributing a lot. Thank you to all of you for listening. Again, I'm very thankful for the funding to present it here. Thank you very much.
Questions?
Nice work. So you mentioned that the catalytic residues were more frustrated. Was that done in the presence or absence of the substrate?
We took all of the proteins with or without substrate. I mean, actually, it doesn't matter because substrates, they are not parameterized in the force field. So you can always say that you are always analyzing without a substrate.
Okay, but if you had the substrate there and you had a parameterized substrate, would that make those residues less frustrated?
Yes.
Okay.
Yeah, hello. Thank you very much. So you've seen that you have two shells actually of frustrated residues. Do you have an hypothesis about why and why you would see this pattern?
Well, it was something that we were not expecting because actually we're expecting the second shell to be minimally frustrated. So no, we don't really have an explanation. It's just what it is. I mean, you see the enrichment of contacts in the two shells, the first and the second one, and yeah, it was surprising to us that the second shell is also frustrated. So we don't really know yet.
Okay.
Maybe a technical question, maybe I misunderstood. I understand, of course, the concept of frustrated residue, but you also mentioned frustrated contacts.
Yes.
What are frustrated contacts?
So frustration is a measure for contacts. So you have your contact and you compare the native energy compared to the distribution of alternative conformations or identities that you can generate. So then, in order to get the frustration index for residues, you need to aggregate all of them together into a single value that can be misleading because if you have a bimodal distribution, then you can see that the single residue is neutral, but actually you have a lot of highly frustrated interactions and minimally frustrated interactions. So frustration is a property of an interaction, and then you can aggregate it into single residues. But usually we like to see interactions because you can get fooled by the single residue. Actually, this is what happened, for example, for the catalytic site 770 in Lactamases. It's neutral, but if you calculate the frustration for the contact map, then you can see that it has frustrated interactions with all the rest of the catalytic sites. So they are participating in a frustrated interaction, but not with the rest of the residues in the protein structure.
Thank you.
Hi, thanks for the talk. So, am I right that the frustration of the residue should be somehow related to the B factor in the PDV structure? And could you comment if you have investigated that?
It could be. We have tried. We had some attempts to try to see if the beta factor is really. Because beta factors can be also linked to conformational diversity, which is Alex is here, and we always try to do this in collaboration. I mean, I don't have the answer. We have done small calculations, but I cannot see properly. Yes, it is.
Okay, thank you.
Maybe next time.
So you touched on the evolutionary aspects at the end. I'm just wondering about, you talked about the globins, but more generally, are more ancient proteins, do you see any change with respect in frustration with respect to the age of the known age of the protein from an evolutionary point of view?
So this evolutionary part is just starting. Actually, we want to extend it to as many superfamilies as possible from the CAF database. But also there are many other things that we can do. For example, I'm really interested to do this kind of studies in resurrected proteins, for example, when you're talking about ancient proteins. The problem that we have is that the protein structures are very scarce. So we need an alignment, and we need a structure for every member, and we need at least 30 structures non-redundant. So, I mean, we can do a lot of questions, but it's difficult to get good data sets. That's why I begged Christine to give us the data, because they have it. And yeah, I mean, also the question is, can we answer these questions with homology models? But they need to really be very high quality. So we are just starting with this, but I'm sure we can study many, many cases, actually.
Okay, thank you. Let's thank Gonzalo again.
Thank you.
Our next